This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 155, recorded on June 22nd, 2017. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello. How are you?
Nice to hear you again.
It was nice seeing you in New Orleans.
It was.
Was it New Orleans? Yes.
It was New Orleans. Yes, it was a lot of fun. We spent some good time together.
We did.
It was a fun TWiM with Arturo, right?
Oh,
yeah,
it was mind-blowing, as they say.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
And from Ann Arbor, Michigan, Michele Swanson.
Hello.
I wanted to get you all in because, that was a fun TWiM, and Arturo was really provocative, and we had a great audience. Couldn't ask for more, right?
No.
It was a good meeting all around.
And I thank all of you for participating as well. I appreciate it. I know it takes time. And
we couldn't do it without you, Vincent.
Thank you. I hope you enjoy it enough and get something out of it as well, because clearly, as you see, we have lots of fans who really appreciate it. So it's great to see that. I want to tell you about the American Society for Microbiology grant writing online course. Learn the best practices in grant writing during this course. You can participate to receive an overview of the funding landscape. You can learn what makes grants successful, receive personalized feedback on your grant proposal. It is an online 7-part series, so you don't have to go anywhere. You can take it right at your computer. It's going to start in the fall of 2017, and there is a registration deadline. You have to register. The deadline's July 5th. It's coming up. And you should go to bit.ly/ASMGWOC1717, bit.ly/ASMGWOC17 to find out more. I also want to tell everyone that I have another book here to give away. Stay tuned to the end of the episode, all right? To find out, you'll have to listen, or I know you can fast forward, but
it's mean.
It's mean. That's the old radio strategy, right? Stay tuned for more of this conversation. All right, we have a snippet and a paper for you today, and we'll start with our snippet, and Michele is going to tell us all about Flint, Michigan.
Yeah, this is work that I presented in a poster at the American Society for Microbiology meeting just a couple weeks ago in New Orleans, and it's entitled Legionella pneumophila in the Flint Water Supply, a Molecular Epidemiology and Virulence Assessment. And the authors include Brenda Byrne, who's been the lead researcher in my group, Sean McMurray, who is at Wayne State, Paul Kilgore, also at Wayne State, Rick Sadler at Michigan State, Nancy Love, an engineering faculty member here at Michigan, and then a whole team of researchers and community activists that have been recruited from our universities, but also from the city of Flint. And this was motivated by an outbreak of Legionnaire's disease that occurred in the summer of 2014 and 2015. So in each of those summers, there were about 40 cases of this atypical pneumonia. And that was unusual because typically there are only maybe 3 or 5 cases each year. So the outbreak also coincided with a period when the city was being led by an emergency manager that had been appointed by the governor. And that's because the city of Flint, it's a proud city that's really had been built because it was a manufacturing base for the big 3 automakers. So there were a lot of terrific union jobs there. But as our manufacturing practices have changed and jobs have moved away and unions have lost strength, there's been economic distress in the city, a declining tax base. The city services have been hurt as a result, and that's part of the reason why the city was under an emergency manager during this period. So at the same time, as a cost-cutting move, they switched their municipal water source. They had been getting their water from Lake Huron, and it had been treated by a Detroit utility, but they switched to the Flint River as the source and had the water treated locally. In addition, for reasons that are not at all clear, the decision was made not to add orthophosphate to the water during the treatment. Now that's relevant because orthophosphate is added to prevent corrosion and to promote the buildup and maintenance of a kind of a shell of metals on the inside of pipes that would prevent the pipes from corroding and keep the water clear. So because orthophosphate was not added to the water, many of the distribution pipes corroded, and there was a large increase in lead in the water, also iron, and then for a number of reasons, including the high iron levels, the amount of free chlorine declined. So there was a massive change in the chemistry of the Flint water source that coincided with this spike of Legionnaire's disease. Now that may be relevant because Legionella is a waterborne pathogen. It lives normally in freshwater and built environments. It forms biofilms, including on pipes, and then amoeba come along and feed off the biofilms, and they've provided a selective pressure for Legionella to actually grow inside phagocytes like amoeba in the environment or macrophages in our lungs.
Are the amoebas influenced by the Legionella? Does the Legionella kill the amoebas?
Well, amoeba have a defense mechanism to protect themselves. They do something called encysting, and they spew out the Legionella in these blebs. So some amoeba are killed, others can shed them. It's beyond my understanding, the complexity of all that biology. But needless to say, there's good evidence that amoeba have provided selective pressure for Legionella to be able to avoid being digested by phagocytes. So it can actually replicate in amoeba, or it can replicate in the white blood cells that patrol our lungs. But you need man-made equipment to throw contaminated water into the air, and then if we inhale it, you can become ill if you don't have a robust immune system. So there was a lot of political pressure then to try to understand why the city of Flint experienced this outbreak, and it was compounded by the year-long delay in notifying the people of Flint that there was a Legionnaire's outbreak going on. So this has been just a really difficult environment, and it meant that our research team needed, number one, to establish trust and communication and credibility with the residents of Flint so that we could go in and do surveillance. So Sean McMurray at Wayne State led this effort and assembled teams of 3 that went door to door in Flint. So a social worker, a infectious disease specialist, and then an environmental engineer would go into the home and conduct a health questionnaire, a needs assessment, give them advice about where they can find services if they're needed, and then also to collect water samples from the kitchen sink, from the shower, and the hot water heater.
Now, Michele, before you go on, were these folks using their water? Because I know after the disaster that happened when they discovered all the lead, a lot of the folks went to using bottled water, and they would only reluctantly use the water in their homes. So were they using their water when you did the survey?
So that is 188 homes were surveyed. And so in some cases, exactly the situation you described, Michael, was the case. In other cases, people maybe used the water more. But we have that data as well. But your point is a good one, because if the water is not flowing because it's not being used in that city, in that home, or if they live on a block where many of the homes have been abandoned, there's less water flow to that region, and that all favors the growth of biofilms, which could increase the bacterial load. So these are all really important issues. So once Legionella was isolated from these water samples, they were sent to my lab. And I'll just say that about 12% of the homes that we surveyed in the city of Flint did have Legionella, and the same percent, about 12% of our homes in our control adjacent communities who had water from a different source also had Legionella. But I want to emphasize that our surveillance campaign took place a full year after the last outbreak, and it was after the city had switched back to their original Lake Huron as their water source.
If you did a random sampling of communities across the U.S., would you find around that much?
So that's a great question, and there has been very little funded research to do that. There are only a few studies. The numbers are not great, but they range around 20 to maybe 30% at most. Homes have been colonized by Legionella. So we're within that range. But again, this was a full year after the outbreak and after the city had already switched back and added orthophosphate so that the water system had started to heal.
That the growing Legionella in the lab is non-trivial. It takes an experienced lab to do it right, doesn't it?
That's true, and that's also true of clinical microbiology labs, and that'll become key as I tell the story of what we found. Any other background questions about the surveillance or the motivation for doing this?
Nope.
No. Okay, so we ended up with about 18 Legionella pneumophila isolates from people's homes, mostly in Flint, but a couple from the control communities. And we also were able to get, through our partnership with the Michigan Department of Health and Human Services, 33 clinical isolates that had been submitted by hospitals in 3 different counties in southeast Michigan. So not only the county that is home to Flint, but also 2 neighboring counties over a 3-year period. So we ended up with a total of 51 strains, both clinical and environmental. And the first question we asked was, are they clonal? Are they in the same family phylogenetically, or are we going to see different clusters, different families in that collection? So to answer that question, we used a method that has been applied by the international Legionella community for decades called sequence-based typing or multi-locus sequence typing, where we amplified from each strain fragments of 7 different genes. We sequenced those fragments and then submitted that sequence to a database. The database then matches the sequence against the whole collection of other alleles that people have been submitting from all over the world, and they match it and give us a number for that allele. So we end up then with a string of 7 different alleles, and we put that together, and it's called the sequence type, or basically the fingerprint of that strain. Now that's useful because we can then look at the sequence types in our collection of environmental and clinical isolates and ask, what's the relationship? Are they all one big family? Are there independent families? What's the story? We can also take that sequence type and then query the literature and ask, has this sequence type ever been found before? Is it risky to humans, etc.? So what we found is that most of the clinical strains formed their own distinct clonal complexes. So we got about 3 or 4 different clonal complexes. And then the environmental samples, the majority of them, formed their own clonal complex. We did have overlap with one sequence type. So sequence type 1, we had 4 clinical isolates and 1 isolate from a home that were the same sequence type, so the same fingerprint. So let me talk about that sequence type 1 first, and then I'll end with describing the environmental isolates. So sequence type 1 globally is the most prevalent of all the Legionellas that have ever been studied and isolated. They've been found in patients. They've been found in environmental surveillance campaigns when there have been outbreaks. They also, here locally, were isolated from patients at one hospital in Flint and also from the pipes at that hospital. So there's great interest in this sequence type 1 as a tool to try to understand whether patients got sick at the hospital or whether they came to the hospital having been exposed in their neighborhood. So that work is ongoing, and it's been in the press. To resolve that question is going to require some classical epidemiology, looking into the case reports, and also more detailed whole genome sequence data to see if they really are part of the same lineage or not. So that work is ongoing. My lab has been most interested in the environmental isolates, and what's surprising about this is that 16 of the 18 were of a different serogroup than is most commonly isolated from patients. So some 80% of Legionnaire's disease is caused by Legionella pneumophila serogroup 1. The majority of the environmental isolates we found in homes in Flint are serogroup 6, so a different surface structure. We also learned that they're either sequence type 367 or a close cousin, sequence type 461. And when we probed the literature to ask what was known about those two sequence types, we discovered that, in fact, they had been isolated from patients in Spain, in Greece, and in Israel. So there was reason to be concerned that they may be put the residents at risk. So our next effort then was to ask, are these strains virulent? And to do that, we started with laboratory assays. So my lab for 25 years has been studying growth of Legionella in macrophages, and we use as a model macrophages derived from the bone marrow of mice. So we can do microscopy and actually look at the outcome of the infection, and we can also do quantitative assays and enumerate colony-forming units as a way to see how well these strains infect and grow in macrophages. And what I'll say is that all our laboratory assays, whether we're looking just at infection the first hour after hitting the macrophage, or if we're looking at growth curve assays that go out 3 days, we're seeing that the environmental and clinical strains are essentially indistinguishable in their ability to get into macrophages, survive, and then replicate.
That's not good.
No, it's not good. So based on those laboratory assays, we can't say that this is not a risk. There is another step of the pathogenesis, of course. In order to get out of the pipe water and into the lung, the microbe is going to have to survive for some period in a water aerosol, either because you inhale it, maybe in the shower, or possibly aspirate it from drinking. So it's formally possible that these serogroup 6 strains can't survive aerosolization as well as serogroup 1. That's possible. We haven't tested that, nor has that been tested in the literature, but that remains a possibility. So you might ask, well, in the clinical surveillance, was any serogroup 6 picked up? And the answer is no, but I have to qualify that because, as I said, some 80% of the Legionnaire's disease is caused by serogroup 1. And therefore, we have a really good, quick, cheap diagnostic test called the urinary antigen test that can readily detect serogroup 1 Legionella. The problem is it cannot detect serogroup 6 Legionella.
So it doesn't see it at all.
I have a figure on the poster showing that 3 different commercial urinary antigen tests need a 500-fold or more higher concentration of bacteria to detect serogroup 6 as compared to serogroup 1. So as a practical matter, it won't be detected.
It doesn't.
Yeah. And in this country, unfortunately, only 5% of Legionnaire's disease cases are confirmed by culture. So we've really, here in the U.S. and abroad, we've become really reliant on this great urinary antigen test for serogroup 1. So altogether, what we've learned is that there are Legionella in people's homes. Curiously, they're serogroup 6, not serogroup 1. They do grow well in macrophages, as well as the clinical strains do. I also want to point out that we do have 2 isolates that were from Detroit, so on a different water system, not exposed to the corrosive pipes that the Flint strains would have been exposed to. And in our laboratory assays, those Detroit isolates are indistinguishable from the Flint isolates. So based on my laboratory data, we cannot say that exposure to the corrosive water chemistry during the 2-year period of the outbreaks led to seeding of homes with more virulent strains. We can't say whether the concentration of those strains was higher during the corrosive pipe period. We can only say, comparing them head to head, they look as virulent, but no more virulent in Flint or out of Flint pipes or from patients. So that's where we are. Currently, we're coming up on the next Legionnaire season. This is a warm weather disease. The bacteria grow better in warm water, so that's when we see outbreaks. So we're going back into Flint and surrounding areas to collect more samples and continue this analysis. And then importantly, we're also going to be able to take the profiles of the water chemistry that is now known from the period during the Legionnaire's outbreak and the period after, and we can expose our laboratory and these environmental isolates to those different water chemistries and then test their resilience to chlorine, for example, or antibiotics or detergents, and also test their virulence in our macrophage growth assays. So that's what my lab will be doing in the year ahead.
Why is it that you don't find the major clinical serotype 1, serogroup 1, you don't find it in the water at all? So
we did find it. We found 2 that were serogroup 1, but that was 2 out of 18. So it's a good question. And it's possible that when we begin to compare the resilience of the serogroup 6 versus serogroup 1 in the more toxic, corrosive water chemistry, that perhaps the serogroup 1s are going to die off and the serogroup 6s will survive. We just don't know that yet, but it's a great mystery that we'd like to solve.
So it's possible that maybe serogroup 6 survive really well in the pipes, but they don't really cause much disease. The 1s might, but they don't survive well, right? That's one possibility.
Yeah, but I should say that serogroup 6 is the second most frequent cause of Legionnaire's disease, and there aren't a lot of studies, but in the studies that have tried to compare severity of disease for serogroup 6 versus serogroup 1, serogroup 6 mortality rates have actually been higher than serogroup 1. So we need to be vigilant.
I thought you said serogroup 1 is the most common one.
It is. And maybe that's because it survives aerosols better. We just don't know.
Oh. So the upcoming season is going to be important, right? Because you're going to...
It is. And I should say all our surveillance, we didn't get funding to do this work until a full year after the outbreak and after they had switched away from that corrosive water, so that they'd gotten the water chemistry mostly under control before we went and did surveillance. So that is a limitation of our study. But we think we'll be able to recreate some of those conditions in the laboratory using these clinical and environmental isolates and try to better understand what happened so that, of course, we can try to avoid a disaster like this in the future.
So, Michele, is the organism that caused the Pontiac Fever, which was the precursor to Legionella that came out of Pontiac, Michigan, hence its name, do you happen to know its sequence type?
So, Pontiac fever, as Michael points out, is peculiar. It's not a pneumonia. It's kind of a flu-like, self-limiting disease that, to my knowledge, they've never isolated viable Legionella from patients with that. It's more like a sick building syndrome where they go back and find Legionella in the water. But I don't know if if Pontiac fever is only caused by specific types of Legionella, specific serogroups or not?
Right.
That's a good question. I don't know the answer to that.
You know, we have a small outbreak here in New York City. There's 6 cases.
Yeah, I just saw that. and of course, the Bronx outbreak was, about 2 or 3 summers ago?
2014, I think?
Yeah, that was a major media market that has put Legionella on people's minds.
Well, thank you, Michele. Good luck with the continuing story.
Yeah, thank you.
We'll look forward to it. At any point in the future, do you get to publish this?
Yeah, we're actually working on that now.
Very good.
Yep. That's the state is interested in getting this information out, so we've had good support from them.
Great. All right. We will now give you a little story about another respiratory pathogen, Coxiella Burnetti, and Elio is going to tell us about this.
Well, Coxiella Burnetti is actually a somewhat distant relative to Legionella.
Yeah.
Both gamma proteobacteria, one a very large group of bacteria, and they are kind of related. So this is a good story. And they're related in other ways also. Coxiella causes a disease called Q fever, which was discovered in Australia. People think it means Queensland because it was isolated. It was first studied there, but actually the Q stands for query because the original finders were so puzzled by it. So it's a disease that has several manifestations. It is very contagious, first of all, and it's said that a single organism can cause disease. Well, maybe, but anyhow, few.
Less than 10, I would say. Yeah.
The disease it causes is a bad flu. You feel miserable. You have a slight fever, and it can last a couple of weeks. It can also develop into an endocarditis and other manifestations. So it's a serious disease, serious enough that the organism Coxiella burnetii has been classified as a possible bioterrorism agent. In other words, people with terrible intentions may think of it as a possible agent of causing disease in their enemy. So anyhow, the story with Coxiella is, I think, quite fascinating because of a couple of features. And one of them is that it has been cultivated in vitro, in laboratory media. And that's amazing because it used to be thought to be a strict, obligate intracellular parasite. In fact, early on, when it was first discovered, it was thought to be a rickettsia, the agents of typhus and other fevers like that, which cannot be grown outside of cells. So what happened was, and Vincent, why don't you tell the story of how this amazing fact was changed, that it can now be grown in vitro, which I, by the way, consider one of the prettiest discoveries in modern microbiology.
Well, I happen to be able to speak with the head of the lab who did this, Robert Heinzen, who's at Rocky Mountain Laboratories over in Hamilton, Montana. And I published it as a TWiM special, I don't know, a couple of months ago. So check that out.
Gorgeous TWiM, by the way. A gorgeous TWiM, if I can say so.
It was very interesting. He told me all about the history and so forth. And I would like to point out that they isolated this at Rocky Mountain Laboratories many years ago, the bacterium, and they found it in a tick. They do random tick screenings there. They collect ticks in the environment and they put them onto guinea pigs. And then if the guinea pig gets a fever, they look in its blood and in its spleen. And in one of them, they were looking for Rocky Mountain spotted fever, rickettsial disease. They found the bacterium of Q fever, Coxiella Burnetti. And he said you can often find it in ticks, but it's not how people get it.
By the way, ticks are amazing in the number of diseases they spread. I mean, Lyme disease.
They are.
Now there's a whole collection of them.
You have to be careful, yeah.
Yeah.
So he said that it's an environmental pathogen, and it's in sheep, it's in goats, it's in dairy cows. In fact, 95% of bottled milk has evidence of Coxiella inside of it. The cows don't get sick, but the sheep and the goats get sick, and it causes abortions. And he said the placentas that are shed at birth have tons of organisms in them. And because infective dose is less than 10 organisms, that's a big source of infecting people. People who work on farms, they get infected at birth by these, when these sheep and goats give birth. And so that's quite interesting. And he said at one point in the dairy industry, there were a lot of people getting sick with this, although now it's decreased. This is an interesting factoid. It's everywhere on the planet. Coxiella burnetti is everywhere on the planet except New Zealand.
No kidding.
Wow.
I always thought that was a safe place to go.
And he said they're trying to keep it that way because they have a big sheep and cow industry there. Anyway, he said,
spread by the respiratory route, right?
It is,
so
yeah.
If the placenta dries and then the soil is kicked up by wind, you could inhale it.
Sure. Yeah, it's a respiratory transmission, and you inhale it and so forth. So ticks have nothing to do with it, but it's environmental, and I suppose it can travel a certain way because you don't need many bacteria to infect you. Anyway, he had someone in his lab years ago who was interested in growing this, and they tried lots of different conditions in the laboratory, and one of the important things was low oxygen environment, and he had a very persistent person who tried all different conditions and got it to grow.
That was actually Anders Olmsland, and he's now faculty at Washington State.
Yep.
But he edited the paper that we are talking about, so he's an editor on the journal.
How interesting.
Frontiers in Cell, yeah. So he'd be a great person to handle this because he really understands the biology.
One of the most startling things about how they got into this, making up this medium that allows the growth of Coxiella, is that they gazed at the sequence. They said, gee, let's think. They had the sequence, the DNA sequence of the bug. Said, let's look at it long and hard and see what genes it has. And from that, they devised a medium.
Yep.
Now that's nice.
And they did a lot of screening, I think, using like biolog technology, where you could screen dozens and dozens of different compositions. So it was really a systems biology approach that
it is,
helped
but
them break the code.
Yeah. By the way, although it can be grown in the lab, and it's been grown in many labs, and this has caused a renaissance in Coxiella research because, think now you can grow it on agar. And it makes tiny little colonies, by the way, about 1/10 or 2/10 of a millimeter, but it does. However, best I know, it does not do this in nature. It is best we know. It seems that it can only grow inside of cells. Mm-hmm. Outside of the laboratory, which is, I think, an interesting point. So, with this in mind, there is a paper that came out with a very tantalizing title. Horizontally acquired biosynthesis genes boost Coxiella Burnetti's physiology. I'll tell you in a second why the title is so amazing. The authors are Moses, Miller, Bonazzi, Beer, and Ragavan. And the interesting part is that some of them are from other places than Montana, but one of the people is from the Rocky Mountain laboratories in Hamilton, Montana, that Vincent visited. And by the way, that's a place where they made many discoveries, including Coxiella Burnetti. So the authors are from Portland, Oregon, from Montpellier, France, and Rocky Mountain. Now, what's so special? Well, what's so special is, think about it. They're talking about horizontally acquired genes. Now, this is a bug that best we know in nature is a strict intercellular parasite. So where does it get the chance to interact with other cells and swap genes? As I'm fond of saying, they're like being in jail. Do they have conjugal visits? So how is it? So the thought is, of course, that this happened before they turned into strict intracellular bugs. They are related. The closest to Coxiella is organism Coxiellas, which are grown in ticks. They do grow in ticks, and these are non-pathogenic. In fact, they are probably endosymbionts that provide the host cells with some nutrients. So they are derived from that, but it must be that the horizontal exchange happened way back, way before the Coxiella went into ticks, and certainly way before the tick Coxiella became the Q fever.
Could I challenge that? So a lot of these bugs, a lot of bacteria are able to take up naked DNA from the environment. They're naturally competent. So many bacteria can pick up naked DNA from the environment, and just because a bug is an obligate intracellular doesn't mean it's not stealing DNA from its eukaryotic hosts. So Legionella, for example, grows in amoeba, and it has a lot of eukaryotic-like DNA. And it's also naturally competent. So I don't think the only horizontal exchange had to happen from bacterium to bacterium before it became an obligate.
Well, let me say this is possible. You're right. Mm-hmm. However, the genes that we're talking about are related to bacterial genes.
Right. They focused their search on other bacterial genes. That's right.
It's true. That's what they looked like. That's right. They may have been looking for the key under the lamppost, but that's what they found.
Right.
So, but you're right. There are other opportunities for bugs to take up DNA. However, I would say that this is not common. Okay, so it's a question. Let's leave it open because we don't know the answer. Here are bugs that have taken up genes, and these genes are of some importance. So there is evidence that there have been. Well, by the way, the paper is interesting in that they were very, very strict in their criterion for what to call horizontal gene acquired, and so for reasons of their own, they eliminated relatives. They didn't compare Coxiella burnetti to the tick Coxiella, for instance, or to Legionella, for that matter. But they went farther afield, and sure enough, they found that these genes we're talking about are really all over the map. They are seen, homologs of those, orthologs of those are seen in a whole lot of different bacteria. Now, about half of them they have about 100 and what is it?
72. 172
genes. So about half of them come from proteobacteria, alpha, beta, gamma, delta, epsilon. But the other half come from gram-positive bacteria. By the way, Coxiella is distinctly a gram-negative. Okay, this will come up. We'll make a point in a minute about why this is important. But so these genes are acquired or are related to bacteria which are all over the bacterial map, even though there seems to be a tendency for their origin being from taxonomically or phylogenetically closer organisms. And this is sort of a general rule of horizontal gene transfer. It appears that horizontal gene transfer took place mainly among related bugs. That is, the more related, the greater the chance of horizontal gene transfer, which probably makes good sense. Anyhow, so what are these genes? Anybody have anything to say about this so far?
Nope, I agree with you in the sense that you effectively are going to take contributions from your neighbors. And so if you're in a niche
your cousins more than your neighbors.
Well, no, neighbors too.
Yeah,
I think
actually,
that's
you're
how
right. Your neighbors is good.
I think that's how it got the gram positives in there.
Okay.
Elio, couldn't these genes have been acquired before Coxiella entered a cell?
That's the idea. Yeah, that's the idea. That it probably happened when Coxiella was some free living organism that decided to become pathogenic. And when it did, it lost a lot of genes. By the way, the genome of Coxiella is about half the genome of E. coli or less, about 2 million bases. So it's reduced, which is typical of intracellular organisms, especially strict intracellular organisms. So anyhow, what are these genes? So these genes are involved in fatty acid biosynthesis and in biotin biosynthesis, which are related. And another cluster is involved in making heme. Now, why they need heme, I don't know, except that they cannot take up heme from the outside and apparently cannot make it other than by having the genes to make it. And heme is, of course, important in cytochrome enzymes and stuff like that. So, and there are a few other interesting metabolic genes, but I want to say something about the heme, because something I didn't know. I bet you that Michael knew, because Michael obviously knows everything. So, like that, Michael?
I was tortured as a young graduate student to learn physiology, so I credit them.
You know that of all things, the making of heme requires a tRNA. Now that I never heard of that. I thought tRNA was involved in supplying amino acids to the ribosomes to make proteins, but here is a glutamine tRNA, or glutamate tRNA rather, which is made where the glutamyl tRNA reductase and an enzyme called glutamate semialdehyde aminotransferase, excuse the long words, convert glutamate into the precursor of heme, which is gamma aminolevulinic acid. Sorry about all this biochemistry. The point is they need to make heme. And they make heme using this tRNA. And one of the genes for the tRNA, or the genes for tRNA, are in fact those which are horizontally acquired. So this is about the story. Mutants in this tRNA grow poorly. And so it looks like this gene, which was a set of genes which was acquired apparently by horizontal gene transfer, is essential and has become essential. And the authors say, by the way, this could be a target for anti-Coxiella therapy because it's a very unique set of genes, so of enzymes. So these genes may be, or the products may be a target. Anyhow,
I have to give them credit that one of their lines of argument was they made a mutant in that tRNA, and those of us who work with E. coli or some other very easy organism to manipulate would not even think twice about that. But this is, very new stuff to be able to make a mutant in Coxiella. It's very
that's
exciting.
facilitated by the discovery that the bug can be grown in vitro.
Now they can do genetics and complement it.
Well, the genetics piece, I talked to the two co-first authors, Abe Moses and Jess Millar, and it was really a active collaboration between those two. And that was the greatest contribution from Paul Baer, who is at the Coxiella pathogenesis section at the Rocky Mountain Labs. He made those mutants for him because what I didn't know at the time is that when you grow Coxiella xenically without the help of the human cells, it has an 18-hour generation time. Yeah. So that is absolutely incredible. If you're a cloner, most cloners are hyperactive. They literally come back and check their plates every 4 hours to see if they got a colony. But if the generation time is only 18 hours, and I talked to Abe and Jess together, they got up really early because they're on the West Coast, and they got up and they were in the lab at 7 o'clock to talk to me. Abe
my God.
It's 7 o'clock their time, not my time. So Abe is from Kansas, and he was responsible for all of the wet work experiments. Jess is the bioinformatician, and we'll get to Jess in a second. But I want to tell you about Abe. He came to Raul's lab, and this is Raul Rekhaven, who is the principal in this story, in 2013 as an undergraduate. And he said he begged to work in Raul's lab. And so here's this older undergraduate student. Abe's now 38 years old, so 2013 is 4 years ago. So he was 34, and he said he'd work for free. And he was able to get some things done. Now, he's not working with E. coli. He's working with a pretty hard organism to work with. And so he began to learn about the care and feeding of Coxiella, and he got really good at it. He's got these golden hands, and Abe credits that the mutant experiment that Michele talked to would not have happened without Paul. He's got golden hands with the exception of making mutants in Coxiella. And so Abe...
Coxiella, please.
Coxiella. And so Abe is going to move on. He's got his master's degree, and he's going to move on, and he's going to start a PhD program in pharmaceutical sciences. But the good news for Raul is he's going to be working in a lab literally next door.
Wow.
And so Raul isn't too freaked out because he's also losing Jess, and he's losing Jess Millar to Michigan.
Yes.
She's defecting to Big Blue. And I don't know how Big Blue recruits, but Jess Millar, she came to Portland State University, where she's been pursuing this dual master's degree in biology and statistics. This makes her the modern Renaissance woman because she has expertise in microbiology. She can grow finicky pathogens. She knows statistical analysis. She's adept at all this wondrous bioinformatics, and more importantly, she loves epidemiology of infectious diseases. So, Michele, when she comes for rotations, you may want to look.
Yeah, I talked to my colleague Evan Snitkin, who has done just some beautiful work, molecular epidemiology using bioinformatic tools, and he was really excited when Jess decided to come to our program. So she'll have great choices here.
What's interesting about her is she got involved with Abe on this project just to round out a figure, but she ended up working with him for a year, and then Raul came up with, he was very reluctant to give two joint first authors. This is new to his lab. It's pretty commonplace to some of the bigger labs where there's equal contributions, but in a small lab, it's not all that common. So he actually warmed to it, and they actually Jess shares two papers where she's co-first author. And Jess herself is a non-traditional student as well. She got involved in science with a bridge to baccalaureate program where, like many students, she went to a 2-year community college, and then she moved to the final 2 years with a science project in a lab. So that's how they bridge you from the community college to the major, if you will, campus, where you're given a science project in a lab. And she came to Raul's lab and said, Raul seems to be a great taskmaster. Raul, I guess, tries to scare him away because she came to Raul's lab and he says, if you want to work for me, you must go and learn Perl. So she said, okay. So she took an online course, taught herself Perl, then solved one of the lab's problems. Perl is a computer science language that they use heavily in bioinformatics. It's sort of like a rector set. You can use it to build all sorts of things that you need to solve problems or issues. And so she taught herself Perl online by herself, and then she solved one of the lab's problems, brought the solution to Raul, and he says, I think you can stay. And so
nice
story.
Very nice.
So, but they were very complimentary about Raul and the environment that he creates. Both Abe and Jess are especially indebted to him. And when I asked one of Michelle's standard questions, you have a strong memory or of an especially exciting or interesting day in the lab, and it's the tRNA that Elio brought up that did it. Working on the glutamic acid tRNA, and it was so different. It was not in any other proteobacterium. And she remembers saying to Abe and Raul, now this is odd. And it's those moments where you say, now this is odd, and you begin to pull on that thread that you really then say, my goodness, we have something.
And great story.
And that's effectively how this story came about.
I got to ask Vincent, did you feel when you were in Hamilton that the Rocky Mountain Lab is kind of a special place where such interactions are possible and encouraged?
Yeah, it's certainly a special place. It's in the middle of nowhere, so they have to assemble a critical mass of people so that you have someone to go to.
To be said for isolation.
And they have a lot of interesting people working in different areas. And I mean, not just bacterial diseases, viral diseases, prion diseases. So I like it. It's very pretty there. And, in another life, I might have gone there. Who knows?
It's also part of the intramural system. So the funding model is completely different. And
That's
I think
right.
it allows them to tackle some of these really difficult biological questions.
Yeah.
Well, let's sort of finish up with a consideration about how Coxiella causes disease, because with all these studies, we should know something about it, and we do. Well, one fact which I have to mention, which is totally unique in microbiology, is that Coxiella burnetti can survive in acidic lysosomes of cells. Those are vesicles which are made by the fusion of very nasty containing vesicles. They are full of enzymes and, I guess, reactive oxygen species, which can kill any other bacterium.
It's the stomach of the cell, basically.
Yeah,
it's the stomach
absolutely.
of the cell, built to digest.
The stomach of the cell. So other bacteria learn to avoid the dangers of that in a whole variety of ways. There's a tremendous number of strategies possible. But Coxiella says, I don't care. I'm going to live and thrive in this impossible environment. And it's unique. As far as I know, no other organism is known to do that. So it not only grows there, but it grows a lot. And even though, as Michael said, it grows slowly, it grows plenty. So much so that the vesicle that it makes becomes a different sort of a structure, which is known as CCV, which stands for Coxiella containing vacuole. And that becomes so big and so full of Coxiella that it reaches almost the dimensions of the cell. It is that big. And at that point, obviously, it breaks up and so forth. But, and it makes sure that the cell doesn't die by inhibiting its apoptosis mechanism. So it's in the Coxiella's interest for the cell not to die, but although eventually it probably does. Anyhow, that is probably not what makes you sick. So what is it that makes you sick? Bugs, being good gram-negatives, contain lipopolysaccharide, also known as endotoxin, which for those in the audience who may not know, is a characteristic component of gram-negative bacteria. It's part of the so-called outer membrane. Gram-positive bacteria have one membrane. Gram-negative bacteria have two membranes. And one of the outer membrane is contains this particular compound, which is known as endotoxin because in high doses it can kill animals. In low doses it causes fever. And it's probably the most common reason why people acquire fever when infected with gram-negative bacteria. Anyhow, it turns out that this lipopolysaccharide, this LPS of Coxiella, is different than that of, say, E. coli or Salmonella, in that it's not very toxic. However, that doesn't mean that it doesn't participate in other conditions. Now, Coxiella is a stealth pathogen in that it avoids causing an inflammatory response. And so it is not clear what causes the disease. In many, many cases, the disease is not caused by the action of the bug directly, but by it eliciting a unbridled immune response. This is true for a tremendous number of infections, but it not necessarily true here, because Coxiella goes to some length to avoid waking up the immune system. And so we're left with a kind of a blank. A lot is known about. There's really some fine papers that deal with what is known about the pathogenesis. Lots is known about how they're taken up, how they inject material via a thing called type 4 secretion into the host cell. But basically, it's still wide open. The simple question, why are you sick with Coxiella, is still wide open. But I think it's a fascinating bug. It's an important bug. And now that we have the tools to study it, I think it has a bright future. Invest in futures in Coxiella research.
I agree. The other thing I really liked about this study is that they did not set out to say, let's find a new drug target. They asked rather a question about evolution. How did this bug that maybe way back when was inside a tick now is inside eukaryotic cells? But in the end, by following that through a really logical, clever approach, they ended up with a metabolic pathway that is now a potential drug target. So I think that just goes to show you don't know where the next clinical discovery will come from, and we need to keep funding basic microbiology research and other basic research.
Do you think it's on how they were trained? Because the way they deciphered the medium for this microbe is through, if you will, a bioinformatic approach where they looked at what genes it had. And they're sort of saying, what can we learn about this organism that we can use to either help us or get rid of it?
Well, more and more people are being trained to do that. So this is, you and I, Michael, see this as a special thing, because it's not how we grew up. But now I think this is going to be commonplace, that there's bioinformatics and experimental science are going to work together at all levels, and they're not separable. They are
but especially it's valuable for these obligate or difficult to culture organisms where you can't just do a whole transposon library or a beta-gal screen. Bioinformatics is going to be a real boon.
Now, I go back to saying what I said before, that I think this is pretty work. And I can't think of anything more laudatory of researchers to say it's pretty.
I agree. I
think it's unfortunate that when you write an NIH grant proposal, you have to write about.
Say that.
You have to write about.
I do pretty research.
You have to write about the health relevance of your work, and people speculate that we could get new antivirals or anti this or that. And I think it should take it out. Just let people find out interesting things, and as Michele said, you'll eventually get something that can be used for health. But to ask you to speculate, I think, is ridiculous.
And if you want to hear more about that, listen to our last TWiM. Arturo Casadevall goes on and on about it.
He did. It was good. All right. Thank you, Elio.
Of course.
And thank you, Michael, for talking with them. It sounded like it was a lot of fun.
It was a pleasure.
Good. I have one email to read from Natasha, who writes, Dear TWiMers, I am relatively new to the TWiM podcast, but it is greatly entertaining and informative. Your recent podcast mentioned an article that was behind a paywall, namely Microbiology of Crocodile Bites in Far North Queensland, Australia. Alas, I can't attach the PDF to this web email, but I have cut and pasted the main text below for your interest. The table containing information about 6 patients where microbiology samples were actually collected doesn't cut and paste very well, but it was interesting that crocodile bites, all saltwater crocs, the most dangerous, were split evenly between wild crocs and farmed crocs. Microbiology from patient 1, P. vulgaris, C. cosseri, group G. strep, patient 2, C. albigans, thought to be contaminant. Patients 3 and 4, B. cereus, also thought to be a contaminant. Patient 5, Staph aureus, patient 6, P. aeruginosa, and Enterococcus species. Although I live down south in Melbourne, Australia, no wild crocodiles here. The newspapers in the top end, Northern Territory, and far north Queensland often have stories about crocodiles and crocodile attacks. Only when a human, usually a tourist, gets attacked and taken, i.e. eaten, do we hear the headlines down south. It would have been great fun to come past at ASM Microbe to listen to you guys in New Orleans, but alas, my conference leave isn't until later in the year. Keep up the interesting work.
Thank you. Nice story.
All right, so that's TWiM 155. You can find it at Apple Podcasts. You can also get it on any of the apps you use to listen to podcasts on your smartphone or tablet. Just subscribe, and that helps us do better for advertising. The more subscribers we have, the more we can get advertising to help us develop this show. If you really like what we do, consider supporting us. You can go to microbe.tv/contribute, and we have different ways you can help out, including a Patreon account where you could give as little as $1 a month, PayPal, and other ways as well. And do send your questions and comments to twim@microbe.tv. I would like to give away a copy of a book called, a brand new book from ASM Press, just received, Food Microbiology by Carl Matthews, Calmia Neal, and Thomas Montville. And this is a lovely book. I really should keep it because it's so cool, but I'm going to give it away because I love our listeners. It's got a section on the basics of food microbiology, growth, survival and death in food, spores, detection. They have a section on pathogenic bacteria, Bacillus cereus, Campylobacter, Clostridium, etc. Listeria is in there as well. They have a section on other microbes important in food, lactic acid bacteria, spoilage organisms, molds, control of microorganisms in food. 4th edition, gorgeous book, lots of illustrations. And how do you get this? Well, send an email to twim@microbe.tv. The subject line should say, I love microbes. And I know we'll get lots of entries, and we're going to take them all, and we'll pull one out at random. That's good. Deadline's July 6th. And, while you're writing, you can say hi to us if you'd like. You don't have to. You can just say, I love microbes in the subject line, and we'll pick one at random. That would make it equal for everyone, so you don't have to be the first or the middle or the last to get it, okay? That's twim@microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter is at Small Things Considered. Thanks, Elio.
My pleasure.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Kandian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. See you next time on This Week in Microbiology.